Good morning. In this session, we'll be dealing with the first week development. In the last session, we have completed the fertilization and the effects of fertilization. So before going to the first week development, let us see the three different periods in the gestation, like germinal period, which is seen in the first 3 weeks, and embryonic period, 4 to 8 week, and fetal period, third month till completion of pregnancy. So in the first week, that is after the fertilization in ampulla, there will be successive cleavage divisions, formation of morula, formation of blastocyst, and implantation of the blastocyst. Overall, it is from fertilization to implantation is what takes place in first week. So repeated segmentation of the zygote is called cleavage division. So this occurs within the zona pellucida by mitotic divisions. It results in small cells known as blastomeres. The large size of the zygote is brought down to normal size, results in normal size of the cell by progressive division of cytoplasm. So in humans, it is holoblastic cleavage due to less deutoplasm, and it is also called total cleavage. And in reptiles and birds, it is meroblastic cleavage or partial cleavage due to abundance of more deutoplasm. And you can see here there is a first cleavage division resulting in 2 blastomeres. Here you can see 3 blastomeres, 4 blastomeres, and 8 blastomeres. Still you can see the zona pellucida is intact, which prevents the implantation at abnormal sites. And you can also see these are the two polar bodies. So stages of cleavage division: 2 cell stage 30 hours after fertilization, 4 cell stage is seen 40 to 50 hours after fertilization, 12 cell stage is seen 72 hours fertilization. It is after this the zygote reaches the uterine cavity. Still it is covered by the zona pellucida. And 16th cell stage is called as morula, which is seen around 96 hours after fertilization. You can see here the rupture of the ovarian follicle, release of secondary oocyte, fertilization. You can see the male and female pronuclei. You can see the two polar bodies. You can see the first cleavage division completed, 2 cell stage, 4 cell stage, 12 cell stage at 72 hours. You can see 4th or 5th day. You can see by 6th day implantation is taking place. At 16th cell stage, blastomeres undergo compaction, adhere to each other by tight junctions due to appearance of E-cadherin, which is an adhesion glycoprotein. From here, cell diversity begins. It is mainly due to OCT4 and NANOG. If these two transcription factors are expressed, the blastomeres will differentiate into inner cell mass, which forms the embryoblast, which gives rise to tissues of the embryo proper. And if these two transcription factors are prevented from expression, it differentiates into outer cell mass, which forms the trophoblast and chorion for protective and nutritive functions. Still, the morula is enveloped by zona pellucida in order to prevent the implantation at abnormal site. You can see here 2 cell stage, 4 cell stage, 16 cell stage covered by the zona pellucida. So 4th or 5th day after fertilization, we can see the blastocyst. The outer cell mass will form the trophoblast. It will acquire epithelium-like tight junctions due to E-cadherin. Then the basal part of this trophoblast is poralized. It pumps in sodium followed by water. Ultimately, a fluid-filled blastocyst cavity is formed. Here you can see a fluid-filled blastocyst cavity is formed. Embryoblast is attached to one side of the cavity, forming the embryonic pole. This will be the embryonic pole, and this will be the embryonic pole, and the trophoblast covering the embryonic pole. This is called as polar trophoblast, and the one which covers this particular part, remaining part, is called as the mural trophoblast. Still, it is covered by the zona pellucida. So you can see here, single layer trophoblast forms the rest of the cavity and forms a embryonic pole opposite to the embryoblast. Polar trophoblast is the one which covers the embryonic pole, and mural trophoblast is the one which covers the rest of the trophoblast. You can see by 5th day, blastocyst has 107 cells, 8 cells are embryoblast, 30 cells are polar trophoblast, and 16 cells are mural trophoblast. The protein synthesis up to each cell stage is by maternal mRNA. After this, embryonic genes will become active for protein synthesis and maternal mRNA is destroyed. Blastocyst is formed within the zona pellucida, approximately 32 to 64 cell stage. You can see here polar trophoblast, mural trophoblast, blastocyst cavity, embryoblast. So outer cell mass and inner cell mass. Implantation of the blastocyst takes place in 6th or 7th day after fertilization. Endometrial changes to decidua. This is called decidual reaction. The major change in decidual reaction is stromal cells will become swollen due to accumulation of glycogen and lipid droplets. There will be 3 zones: stratum compactum, spongiosum, and basal. Zona pellucida disappears by the end of the 5th day due to trypsin-like enzymes, action of trypsin-like enzymes. Then polar trophoblast implants into stratum compactum and spongiosum, because these two layers are eroded by the proteolytic action of the trophoblast. Then L selecting of the trophoblast adheres to the carbohydrate receptor of uterine epithelium, and the integrins from trophoblast will interact with extracellular molecules like laminin for attachment and fibronectin for migration. These molecules also help in differentiation of the trophoblast. You can see here, this is a polar trophoblast, mural trophoblast, embryoblast. You can see the trophoblast invading between the uterine epithelium. So now here you can see there is two types of trophoblast, cytotrophoblast and syncytial trophoblast, which will be more prominently seen in the second week. You can also see a bilaminar germ disc, which is also more prominently seen in the second week. This is the transition, that is seventh day. There is a transition from first week to second week. You can see the implantation is half complete. You can see this is the embryonic pole. This will be the embryonic pole. You can see the amniotic cavity covered by the embryogenic cells. Here you can see the implantation is complete and the site of implantation is closed by the fibrin plaque. This green one will be the syncytial trophoblast and this will be the cytotrophoblast. You can see the amniotic cavity, primary yolk sac. You can see the bilaminar germ disc. That means it's already in the second week. Now, site of implantation, junction of fundus and posterior wall of the body of the uterus is the commonest site for implantation, but anywhere in the upper part of the uterus is considered normal. So, abnormal implantation, ectopic pregnancy, that is extrauterine pregnancy. Anywhere outside the uterus is ectopic pregnancy. So, common sites will be ovary, uterine tube, and abdominal cavity. The conceptus cannot survive. But when the ectopic pregnancy is within the uterine tube, the most commonest problem is uterine tube lumen is very small. It will cause rupture of the uterine tube, which is an acute emergency due to severe bleeding, which will have the mother at risk. So immediate surgical intervention is required for this. Then intrauterine abnormal implantation is when implantation occurs in the lower part of the uterine cavity, which results in placenta previa, because this results in severe bleeding during delivery or in the later part of the pregnancy. Thank you.